My name is Vamsi. I'll be leading you through the ChIP-Seq features in Avadis NGS. Avadis NGS has been designed for biologists, which means it's been designed to insulate users from the complexity and size of NGS data by hiding the complexity behind easy-to-use workflows and visually appealing windows into the data. For those of you who are familiar with GeneSpring, the user interface is very similar to that in GeneSpring. Several thousand researchers around the world use GeneSpring, so the learning curve should be quite short. Even for those of you who haven't used GeneSpring, we believe that Avadis NGS will be quite easy to use. So the main question that a ChIP-Seq experiment tries to answer is, what does transcription factor X do in a specific condition? In other words, we try to answer the question, what genes are affected by the transcription factor X under a particular condition? This in turn we formulate as two computational problems. What genomic regions does X bind to? And that is solved by the peak detection algorithms. And secondly, what genes are adjacent to the binding regions? And this we infer using the annotations that we package with the tool. So given these two computational blocks, we'll be able to predict what genes are affected by the transcription factor. And after that, there can be two different types of analysis. You could be interested in, why does the transcription factor affect only those genes? In other words, what is specific about the DNA elements to which the transcription factor bound? That is the motif detection problem. Or you could be interested in downstream analysis. What processes are affected by the transcription factor? For this, we would simply take all of the genes that are affected and carry out Go analysis, pathway analysis, etc. Another question could be that under this particular condition, we know what are the genes that are being affected, but what other genes could potentially be affected by this transcription factor? For that, we can take the motif that it binds to and scan the entire genome and find other occurrences of the motif. So all of those other genes are also potential candidates which could be regulated by the transcription factor. Okay. So first, we'll go about how do we create an experiment in Avadis NGS. To create an experiment, we first specify the experiment type, which is currently one of ChIP-Seq, RNA-Seq, or DNA-Seq, and that is there in the top dialog box. And after that, we specify the metadata for the experiment. We need to specify the organism of interest and the genome build for this organism. We provide prepackaged annotations for a variety of organisms, and they can all be downloaded within the tool itself. If you do not find a bundle for your organism of interest, then you can let us know, and we are always happy to create new annotations on demand. After that, you need to specify the sequencing platform from which the data was obtained, whether it's Illumina, Solid, 454, Ion Torrent, etc. A number of special steps have special handling depending on the platform. For instance, we have special QC plots for Illumina. For RNA-Seq, strand-specific protocols from Solid, we have special handling. And similarly, if the data is from 454, when doing the SNP detection, we take special care of the homopolymer stretches, where 454 reads tend to have more errors. Finally, you specify whether the library layout, whether it's single-end or paired-end sequencing. ChIP-Seq data is usually single-end data and does not make any use of the pairing information. After that, we need to import the samples into the experiment. Standard file chooser will open up. You can choose files from your file system. You can specify SAM, BAM, or ELAND export files in the system. For files which have a long name, you can provide alternative, nicer sample names, because these are the names that will be displayed in the navigator, in the genome browser, and other locations. If your sample is split across multiple files, You can also combine the multiple files into a single sample by providing the same name for all of those files. After this happens, the specified files will be imported into the system. When the import finishes, you will see the above view in Avadis NGS. At the top left, you see the project navigator, which shows the organization of work into projects and experiments. In this case, the experiments folder is not expanded out, but in general, a project can have multiple experiments, even of different types, DNA, RNA, etc. And each experiment can contain many samples. One project is open at a time, and the experiments in that project are shown in the project navigator. In this example, a single experiment is open in the navigator, and the name of the experiment is NRSF/REST. You can open multiple experiments. Results can be shared across experiments, and that is quite useful since ChIP-Seq and RNA experiments are often performed together, in which case you would most probably create a single project, have the ChIP-Seq and the corresponding RNA-seq experiments within that project, and share results between those two experiments. On the right, you see the workflow containing the common analysis steps for this particular experiment type. Avadis NGS has specialized workflows for DNA-seq, RNA-seq, and ChIP-Seq analysis. At the bottom left, you see the experiment navigator containing all the results for an experiment. Since we have just opened this experiment and imported data, the navigator is sparse. It only contains the reads that have been imported and the details of the samples. But as you continue to do your work, the new results like read lists, region lists, gene lists, clustering trees, pathways, all of those will be created in the navigator in a logical manner. And the hierarchy contains the audit trail of those results, placing, say, a gene list obtained using a particular read list as an input as a child of that particular read list. Each time you run a particular operation from the workflow, you will need to specify one or more objects from this navigator as inputs. The result of the operation will be added back to the navigator at the appropriate place. Finally, in the middle of the tool, you see the genome browser, which allows you to view all of your results against the genomic backbone. The genome browser shows reads, genes, transcripts, SNPs, continuous data like coverage and conservation tracks. Any of the result objects that are created in the navigator can be dragged and dropped into the genome browser, and it will create a special track with the visualization appropriate for that particular object. Since we just started the experiment, all that we have in the navigator are the samples. You can double click on a sample. You can double click on a sample and it will launch the sample inspector. So this dialog shows the number of reads matching against the various chromosomes in the build. In many experiments, we expect the number of reads to be uniformly distributed across the genome. So larger chromosomes will show a higher read count than smaller chromosomes. And in ChIP-Seq experiments, we also expect to see approximately equal number of reads from each strand. So these kind of sanity checks can be quickly performed by going over the numbers shown in the sample inspector. So Avadis NGS provides multiple QC plots. Some of the QC plots like lane and tile QC are specific to the Illumina platform. Some other QC plots like mate status are specific to paired end libraries. But alignment score, mapping quality, distribution QC plots are common across all platforms and experiment types. Due to time constraints, we will not be exploring the QC plots in this presentation. However, in a typical analysis, you would consult the QC plots and arrive at filtering criteria. The most common filtering criteria are all available in a single filter workflow, filter by read matrix. The dialog box of filter by read matrix is shown on the top right hand corner. So you can specify multiple filtering criteria, and only reads which pass all of those filtering criteria will be created as a child read list. For ChIP-Seq experiments in particular, it's highly recommended that a second filter which removes duplicate reads be carried out, since most peak calling algorithms assume that duplicates have been removed prior to analysis. So now that you have inspected the samples and removed the bad reads, the next step is to predict the binding regions. We have three different peak calling algorithms in the workflow. The first is a very simple sliding window based algorithm that is simply labeled as enriched region detection in the center. The second is a very rigorous expectation maximization based algorithm called PIX. And the third is a popular open source algorithm called MAX. PIX and MAX are both published algorithms. We have implemented the PIX algorithm and optimized it to make optimal use of the computing resources available on your machine. In the next few slides, we'll go over each of those algorithms very briefly. So when you click on the peak detection by PIX workflow link, it shows a parameter dialog box. The first parameter in that is the read length, which has been automatically inferred from the sample that was loaded. The second parameter is the fragment length with which the experiment was carried out. The default value shown here is 200, but you can modify it depending on your experimental conditions. PIX will then find all of the regions in the genome which have a large number of reads. For each one of those regions, it analyzes the pattern of positive and negative reads to see if the pattern of those reads is consistent with the presence of a binding region. In general, when there is a particular transcription factor binding site, you would expect to have a large number of positive reads to the left of the binding site and a large number of negative reads to the right of the binding site. And the distribution of these reads is expected to follow the normal distribution, and that is the picture that is shown at the bottom. So PIX evaluates each region which has a large number of reads to see whether the distribution of reads is consistent with the pattern that is there in the picture at the bottom. And regions which satisfy the criteria are all saved as a region list. As I mentioned earlier, the version of PIX that we have implemented is a parallel version, so it makes use of all of the computing resources available on your system. The output of the PIX analysis, the peak detection analysis, is a region list. The region list is by default labeled peaks detected by PIX, and it is created as a child of the read list which was used for the analysis. You can look at those regions either by double clicking on the peaks detected by PIX region list or by right clicking on it and choosing the inspect region list menu item. When that happens, a region list node inspector is opened as is shown on the right hand side. Region lists are a very fundamental object in Avadis NGS. Each region of a region list has chromosome, start and end, and after that it can have any attributes or properties that are specific to the workflow which created the region list. The PIX algorithm, for each of the binding sites that it finds, it annotates it with score and FDR. So we see a 5-column spreadsheet out here. And as you can see, PIX detected 1,762 potential binding sites. Similarly, we can run MAX with its default parameters. And MAX has created, and the output of MAX is also shown here. MAX has created a region list with 7,497 regions. The parameters with which an operation has been run, all of those parameters are stored in the note section of the object. So at a later point in time when you inspect a region list, you know exactly how that region list was created. You'll also notice that the properties that MAX has provided are different from what PIX has provided. As I mentioned earlier, the enriched region detection algorithm is a simple sliding window-based approach. It can be used for detecting methylation patterns or in general for finding locations on the genome with a large number of reads, but it should not be used for transcription factor binding site detection, which is done much better by the PICS and MAX algorithms. It is shown here for completeness, but in the rest of the presentation, we'll be talking about the PICS and MAX outputs. Since region lists can be very large, there are a number of utilities in the Avadis NGS workflow which help in summarizing the information in a region list, help to prioritize the regions in a region list, and help to compare two different region lists. For instance, now we have two different region lists, one output by PICS and one output by MAX. So in the next few slides, we will see how to compare these two region lists and how to get a feel for what are the properties of the regions in these region lists. For getting a high-level view of a region list, the region list operations utility can be used. This utility allows you to see the distributions of the various properties like chromosome, score, and FTR. Here, for instance, we see that chromosome 1 has the highest number of binding sites, followed by chromosome 19. So this can be used for verifying any hypothesis that you'd have, which is a very high-level hypothesis. For instance, if you thought that the binding sites would be equally well distributed across the genome, then you'd expect the number of binding sites to be proportional to the chromosome length. But as it turns out here, that is not the case. Chromosome 19, which is much smaller than chromosome 2, seems to have a larger number of binding sites. And just as in Excel or other spreadsheet programs, new properties like length can be defined and added to the region list. For instance, if you want to get a distribution of the lengths of these binding regions, length is not a property that PICS has added to the regions. We only have the start and the end. But using the new formula utility which is present in this view, we can create a new property. And now I've shown you a view of the same spreadsheet but with the length attribute added. And after that, we can plot the distribution of the lengths. And we see that the median length of a binding site from the PICS region list is approximately 85. So the region list operation has multiple components. You can draw histograms. You can draw a scatter plot of two properties against each other. You can compute summary statistics like median, 75th percentile, etc. You can create new formulas. In addition, on the right-hand side, what you see are filters. You can change the filter criteria. For instance, you can say, show me only the regions with FTR less than a particular value. Show me only the regions with a score greater than a particular value. All of these filters are applied dynamically, and the histogram or any other view that is shown out here will adjust itself appropriately to the filtering criteria. So as I mentioned earlier, instead of looking at histograms, we can also look at the summary statistics. So now when we look at the summary statistics of the MAX and the PICS outputs next to each other, we see that the median length of regions output by MAX is 284, whereas the median length of the regions output by PICS is 81. So in a sense, the regions output by PICS are much more fine than the regions output by MAX. And this is also an important criteria if you want to do downstream analysis like motif detection, because motif detection is computationally expensive and the time that it takes is proportional to the size of the regions that it's analyzing. Okay. So we now have an idea about the individual region lists. We know their chromosomal distributions, we know the length of the regions, we know the distribution of scores, etc. But what we need to do is to say, do the two results agree with each other or not? For instance, now do the PICS regions overlap with the MAX regions? Do the MAX regions overlap with the PICS regions? Since MAX has generated so many more regions, are there any good regions in those regions or not? For that, we have a utility called compare region lists. So we can compare the PICS regions versus the MAX regions. And this generates 4 different region lists. The first one contains all of the regions of PICS that overlap any of the regions of MAX. The second contains the regions of PICS which do not overlap any of the regions of MAX. The third contains the regions of MAX that overlap a region of PICS. And the fourth contains all of the regions of MAX which do not overlap any of the regions of PICS. So usually when we do a set operation, when we compare 2 sets, we get a Venn diagram with 3 elements in it. But because of the nature of region lists, when you compare 2 region lists, you end up with 4 different region lists. What we are really interested now is when we look at the MAX regions, since there were so many of them, what are the properties of the MAX regions that overlap the PICS regions? And what are the properties of the MAX regions that do not overlap any of the PICS regions? And those are the 2 different summary statistics that are shown out here. The summary statistics shown on the left are the regions that only MAX predicted, and the summary statistics shown on the right are those that were common to both of them. And the score, the median score value has been highlighted in these 2 summary statistics. And as you can see, wherever the regions are in common, they have a much higher score. The median score is 1145. And if we take only the regions output by MAX, the median score is only 71. So in a sense, what this stat conveys at a very high level is that the regions that only MAX detected are of lower quality. Or in other words, the PICS output is has higher confidence. So now let us take a look at the genome browser and how do these different regions look. So the top view of the genome browser is showing you the read track. The top half of the read track has coverage distribution. The positive reads are shown in blue and the negative reads are shown in red. And you see the classical pattern of a positive coverage to the left and a negative coverage to the right. And we see the individual reads below it. And now we can see how the different regions look. MAX has a tendency to output broad regions, and that is shown in the first track. And the second track is a narrow region, and that contains the PICS output. And the last track is the enriched region detection. But enriched region detection is completely dependent on parameters, so let's not spend too much time on that. You'll also notice that the tracks at the top right hand corner, each track has a left and right arrow button. So by simply clicking the right arrow, you can keep going from one region to the next region in that region list. So in the genome browser, you can conveniently investigate one region after the other and look at the read pattern in the top track. The initial problem we were interested in is what are the genes that are affected by the transcription factor. So we solved the first part of the computational puzzle. What are the binding regions for that particular transcription factor? The next step we want to carry out is find out what are the genes in the vicinity of those binding regions. For that, we use a utility called translate regions to genes. When we click on this utility, it will ask for a region list, and we are specifying the PICS region list. It then takes a look at the annotation that is prepackaged with the tool and identifies all of the genes which are within a 5000 base pair distance from the binding site. That cutoff can be changed using the change cutoff, and in this particular case I've changed the cutoff to 1000 base pairs. So now it is showing the list of 839 genes which are within 1000 base pairs of the binding sites. And the distance distribution is also shown as a histogram. Once you're comfortable with the cutoff, you can click finish, and it creates the list of all of these genes in the navigator. So for instance, here it has created an entity list called neighboring genes from PICS regions. One can then double click on that object, or one can right click on it and say inspect list, and we get the entity list inspector. The entity list inspector in the spreadsheet shows all of the genes which have been saved. So we have the 839 genes here which are within a 1000 base pair distance of the binding sites. Once we have an entity list, we can use many of the downstream analysis functionalities in Avadis NGS. Go, GACA, pathway are some of those downstream analysis steps. If we do the Go analysis, then these are some of the Go terms which are enriched. We see synapse, synaptic transmission, transmission of nerve impulse, etc. And all of these results agree very well with the published results for the NRSF dataset. Now that we see terms like synapse and nerve transmission, etc., we can also use the downstream analysis step called mesh pathway construction. For human, mouse, rat, and 4 other model organisms, Avadis NGS makes available an interaction database. The interaction database has been obtained by running an in-house natural language processing tool on all the Medline literature available. The interactions that have been extracted from the natural language processing tool are packaged into organism-specific databases. So we have one database which contains all the interactions that are relevant for humans, another database which contains all the interactions relevant for mouse, etc. The mesh pathway utility allows you to interrogate this database. For instance, here I'm searching for the term synaptic, and I'm choosing the mesh term synaptic transmission, and it says that overall there are 3,800 relations present in the database which are occurring in abstracts which have been tagged with the mesh term synaptic transmission. The mesh terms are assigned by curators at NCBI. So pulling out these interactions means these interactions are all somehow related to synaptic transmission. So once we pull out all of those interactions and restrict ourselves to protein-protein interactions, this is the pathway view that we get. So these are all protein-protein interactions occurring in abstracts which have been tagged with the term synaptic transmission. In addition, I have now clicked on the entity list neighboring genes from the PICS regions, and the common genes are highlighted in this particular pathway in blue. So you can see that some of the genes that we found through our analysis of the PICS regions are actually present, are known to occur in literature, and are mentioned in the context of synaptic transmission. Another way we can perform this analysis is to start from the transcription factor itself. NRSF is also known as REST. So we can start from REST and say what all proteins is REST known to interact with. So we carry out a one-step expansion from REST, and it creates a pathway showing all of the interactions involving REST and other proteins. One can again click on an entity list in the navigator and see that there are quite a few of these targets of REST that are known in literature, which we have also identified using our workflow. When you have multiple entity lists, you can carry out things like 3-way Venn diagrams and 4-way Venn diagrams and identify very high confidence targets, or you may want to go after targets which are not known in literature but which the PICS algorithm has still identified as potential targets. All of those can be carried out by comparing the entity lists. The other stream of analysis that we wanted to do is identify the reason why the transcription factor is binding to those specific DNA regions, and that brings us to motif detection. In Avadis NGS, the motif detection algorithm that has been implemented is called GADEM. GADEM is a published algorithm, and it is a genetic algorithm variation of the ME implementation which people commonly use for finding motifs. GADEM has a large number of options which dictate the speed with which it runs. Since it's a genetic algorithm, it has a notion of population size, that is the number of starting points that it will investigate simultaneously, and a number of generations through which it tries to evolve a solution. A thorough discussion of GADEM will require a separate webinar. So for now, we have just shown you a brief idea of how to configure it. Once the motif detection is carried out, and the motif detection, the time that it takes is directly proportional to the number of binding regions that you start off with. So it is highly recommended to choose the PICS output and maybe only the top 1000 or 2000 top scoring PICS regions for the motif detection analysis. Once that is carried out, all of the different motifs which have high confidence values are output. This is the motif with the highest number of matches, with 1357 matches. And the motif logo is shown in the motif inspector. And the motif inspector has two tabs, one where it shows the actual motif, and the second where it shows all of the regions where the motif occurs. So out of 1700 regions, 1357 of those regions happen to have this particular motif. So it's a very, very strong motif. One can then go and search in public databases like Jasper and see if there is any known motif for this particular transcription factor. And Jasper database does indeed contain a motif. The motif that it contains is shown at the top, and the reverse complement of it is shown at the bottom. And by comparing the two slides, the previous slide and this current slide, you will notice that the motif that has been detected by GADEM is almost an exact version of the motif present in the Jasper database. So that pretty much concludes our overall ChIP-Seq workflow. We started with reads. We looked at some of the common filtering options that need to be done. Then we looked at three different peak detection algorithms: an enriched region detection, the PICS algorithm, and the MAX algorithm. After we find the binding sites, we can either use GADEM to find out a motif, or we can pursue downstream analysis in the form of genes. For that, we saw how to convert a region list into genes. And once we have an entity list which comprises of genes, we can carry out GO analysis, GAC analysis, pathway analysis, etc.